Hello, everyone, and welcome to today's broadcast, Trends and Emerging Applications in Single-Cell Genomics, presented by Stephen Hoffman, Segment Marketing Manager for Single-Cell Sequencing, Illumina. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Illumina. For more information about our sponsor, visit www.illumina.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Mr. Hoffman. I will now turn the presentation over to him.
Thank you. It's a pleasure to be here today. And I'm going to speak today about some of the things that I see as I have been all around North America in a bunch of different laboratories and kind of keeping an eye on what's going on in single cell. One of the things that I think is most important when we think about single cell genomics is why do you want to do single cell genomics in the first place? And to get at that point, I think two analogies are really useful. One is comparing bulk analyses of genomics to a fruit smoothie. While it is a delicious beverage, it can be hard to really know what's going on with any of the individual components of that before they were all blended together. In much the same way, if you look at the cells of an immune system, if you blend them all together, it's impossible to tell which alpha and beta strands were together in a single cell. Whereas if you do that analysis at the single cell level, that's easily sorted out after you look at the sequence data. Another thing that makes a really good analogy for why look at single cell genomics rather than bulk genomics is if you think about what happens when, my 11-year-old son does laundry. We've had an entire load of whites come back as pink on account of getting one red piece of laundry in there. This can happen in situations where one or just a few cells are expressing a gene or a packet of genes at a very high level. And if you look at that in bulk, it appears as though all of them are at a relatively low level of expression and not just a few high expressors. Single cell isn't new. We've really started on the route to single cell genomics way back in the 1660s with the earliest microscopes. But the advent of single cell qPCR in the late 90s really built on the single cell work that started with flow cytometry. As the technologies improved and the sensitivities of the techniques improved, in the early 2000s, we were able to start doing sequencing of single cell RNA and single cell DNA to better understand what's going on at the single cell level. I see a lot of trends as I study single cell genomics, and one of the most striking trends I see is that in 2009 or 2010, even a few years after that, you could get really prestigious journal articles accepted with just a handful of cells, but the trend continues to go up and up. And if you look as this graph goes up and to the right, this is a log scale. So the number of cells that are being reported in individual papers is really going up at a very fast level with the advent of droplet-based microfluidics. It's really very striking how many cells can easily be included in research projects today. Another trend that I see as I've traveled around and read a bunch of papers that are coming out is that there's just an avalanche of new methods. In 2015, 16, and 17, there were many more new methods that come out. And it can be overwhelming to look at all of these different methods and very minor tweaks from one to the other sometimes to understand what is the best method that is available for your particular research. And hopefully as we go through some of the emerging applications today, you'll have a good idea about which approach might work best for you. But if not, we're more than happy at Illumina to help guide you through that process. The other trend I wanted to make sure I mentioned is shown in this slide, and that is there's a trend towards more and more of the studies that include single-cell genomics being multi-omic. And by that, I mean including multiple layers of that central dogma of molecular biology. We've got studies that include DNA and RNA, or RNA and epigenetics like ATAC-seq, and I think most excitingly, to me at least, is the combination of protein detection with whole transcriptome. So as these methods continue to be more widely accepted, we see a lot of the newest papers being multi-omic, either multi-omic in terms of multiple experiments to cover the different modalities, or even now with site-seq and size-seq and a lot of these protocols called out on the bottom rectangle, you're able to do multi-omics in a single experiment, in single protocol. Multi-omics kind of leads to the other trend, which is more and more highly dimensional data. So as the number of cells increases, you look at more genes and more proteins, more epigenetic features, and you start to do things now like we're used to doing with other bulk approaches like multiple time points or time course experiments. The trend overall is to more and more highly dimensional data. And one of the things that brings up is how do you analyze highly dimensional data? One of the most common approaches, and the type of graph that we're going to see in a number of examples later on, is what's called t-SNE. So on the left here, you see all of the different cells in this experiment being clustered together by how similar the profiles of all of these different features. So if they express many of the same genes, they'll be more closely related in that multi-dimensional space. What you do with t-SNE is you take that and you smash it down to a two-dimensional representation. And therefore, the cells that are closest together are the ones most similar across all of those different dimensions of the data. And so a lot of times that will lead to clustering, where in this example, you can see a red cluster, a blue cluster, and a green cluster. So there are basically three subpopulations of cells in this example. Regardless of which of the multi-omic approaches, if you want to do DNA or RNA, whole transcriptome, you want to look at protein or epigenetics, or if you even want to take your system and perturb it through the use of CRISPR or drug treatments or environmental treatments, all of those readouts can be read via Illumina next generation sequencing. And we're going to take a look at the basic process, the general process of how that's done. Regardless of which type of analysis you want to do, the first step in a single-cell genomics project is to do tissue dissociation. And this is actually a place where most of the problems really start. As with a lot of genomics approaches, if you put poor quality samples in, you're going to get lower quality data out. After that first step, then you go through to cell isolation, and then you amplify the DNA, RNA, or the nucleotide signal that is used to signify the epigenetic or proteins. You do a library prep in step 4, get the adapters onto the end of those molecules, and then you sequence it and analyze the data. So there are a number of different places on the web where you can go to find additional information about tissue dissociation. And there are a lot of really great companies out there that will help you with this even more than Illumina does, like Miltenyi or Stem Cell Technologies really use their expertise to help you get good, clean cell isolations. We're going to look at an example of how different preparations can actually affect the data that you get from a single cell experiment. Here, looking at mouse neurons. On the left-hand chart, you can see the difference in, first of all, just the number of UMIs or genes that are detected from cells that are prepared using a GentleMax preparation, which is a combination of light mechanical and enzymatic action versus a straight mechanical preparation. And so if you look at the blue bars, you can see you get higher overall diversity in your libraries when you use this gentle combination isolation. The other thing that becomes important is even the most gentle or the most appropriate isolation can still lead to some cells being killed or stressed. And so therefore, doing a step where you filter out those damaged cells can really help improve the overall quality also. And we'll see an example next that shows some clustering results. On the left are all of the cells from young mouse neurons. And on the right cluster diagram here are all of those cells that were isolated from an older mouse. Now, for neurons, the older the sample source, the more difficult it is to get good separation, and the more likely that separation is to cause some stress to the cells. And you can actually see the difference here because you get a large number of distinct clusters generated from the young neurons. But in the older neurons that underwent the same preparation, a lot of the cells are expressing stress response genes, or they have undergone cell death. And so then when cells die, the proportion of the total reads that go to mitochondrial genes or ribosomal RNA genes goes up. And so what that does is it pulls all of those cells that have different gene expression patterns together because they all have those same features being very prominent in their gene expression profiles when they're damaged. If you take the cells on the left and you include all of the cells, you have this same graph we saw on the last slide. But here, if we color code them, red are all of the cells, and blue is the subset of those cells that you got after you did a filtration. And you can see an entire cluster, as indicated by this blue arrow, that is only present when you do not do the filtering. And since this is a filter that removes dead cells, what that means is that this entire cluster is likely to be those cells that are damaged or very stressed. So a number of different approaches to look at it, but all of them indicate that having good, high quality cells in single cell suspension will end up giving you cleaner, easier to interpret results. Step two in that general 6-step protocol is cell capture or singularization. There are a lot of different ways to do this, and a lot of different partners out in the ecosystem that can help you do that. So pipetting, manual manipulation, laser capture, these are very labor intensive approaches, but they have the advantage of maintaining very often spatial information. And so therefore people are still using these on some experiments or in conjugation with other approaches. The middle column here is the one approach that we've been doing for a long time as well, and that is using FACS sorting to based on usually some sort of cell surface marker of interest to isolate a subset of the cells into a tube or into individual wells before you lyse them and then do your library preps. Droplet-based microfluidics are also a very common and really high throughput and a low cost per cell method. There are a number of providers right now, like Biorad makes a DD-seq instrument, 10x Genomics makes a Chromium instrument, Mission Bio makes their Tapestry. There are more homemade methods or homebrew methods like DropSeq. All of them use oil-water mixtures to isolate single cells in a droplet or a bubble with the reagents necessary. Then there are these nano chamber or microfluidic devices that use some sort of a mechanical or chip type of a thing, a slide perhaps, to isolate individual cells physically. And then you can go through the library prep and amplification steps within that microfluidic environment. Regardless of the capture method that you use, whether it's something that's very low throughput or something like a droplet-based approach that can generate 5,000 to 8,000 cells per experiment, there are different Illumina sequencers to match each of those individual experiments. The technology you use for isolating your cells can make a difference in the type of data you get, though. If you look at this paper from 2017, it compared a number of single cell approaches that used what we call 3' end reads, where only the very end of the read, just above the poly A tail, is read, and this were droplet-based approaches in this example. And they compared the data to some different chemistry approaches like FACS sorting or a microfluidic approach that generated full-length cDNAs, and then you fragmented them and sequenced them. When you take a full-length approach to your transcriptome, they demonstrate here that you get more genes being detected. And that continues to play out in the very recent paper. And this was actually looking at the exact same cell types being isolated from mouse. And you can see the throughput impacts. So when they use a 3' RNA-seq in a droplet-based prep technology, they detected they were able to isolate more than 7,000 cells, and they detected 1,600 genes per cell. You compare that with, the same tissue type. It's lower throughput when you do RNA-seq from FACS sorting. They only got 300 cells, but the number of genes detected per cell is actually much higher at 6,000. And you can see in the bar graphs on the right, all of the histograms that show a little more detail on those average numbers. Wally et al. Had another paper where they directly compared a microfluidic, a low cell number, high sequencing depth, approach with a droplet based, which is a higher cell number, lower sequencing depth approach. And you can see that there are significant differences in the number of cells, the reads generated per cell, 208 cells with 1.5 million reads per cell, and it turns up a higher number of genes than the droplet approach with 15,000 reads per cell, but you're able to get many more cells out. And what I really like about this paper is it shows you the power of more cells. So this shows the grouping of the cells in what are called violin plots, and they're color-coded by what group they're in. So on the top, you can see there's 3 groups, a green, a red, and a blue group. But if you take that droplet-based data where there's many more cells, you get enough cells in the green group to subdivide it into 2 distinct groups, what they call G1 and G4 in the figure. So this kind of shows you that you can really get better resolution of your groups by including more cells in the analysis. And that makes sense to anybody who's done population genetics. The more members of a population you look at, the more likely you are to get good segmentation of all of the subgroups. Regardless, though, if you combine G1 and G4, both of the different approaches group the cells into similar bins. So there's very good agreement between the 2 different approaches, even despite the major difference in the number of genes detected. There's now also one example that I'm aware of, this Hayashi paper, where instead of doing just the poly A transcripts, so that's 7,000 transcripts in this example, by doing a whole or total RNA approach, they're actually able to look at and identify another 450 transcripts in this cell type. And so that's good news as we move forward for people who want to look at long non-coding RNA or microRNAs or all of the other things that are not polyadenylated in a cell. So chemistry used, we saw also certainly impacts technology, but also the actual sample in an experiment can affect the sensitivity. This Bagnoli et al. Paper, it actually is a paper where they were talking about a new method that they call molecular crowding scrub-seq, and it seems to be very, very sensitive. But one of the real gems in this paper is that they have this comparison of all of these different experiments that included ERCC controls. So these are known artificial RNAs. And you're able, you know how many of them go into the master mixes. So you've got a theoretical detection or a theoretical concentration. And then you can compare that to the actual counts and you can see what is the probability of detection. And the blue arrows and the green arrows are the same technology, or all the blue arrows are the same technology, and the green arrows are the same technology. But you can see significantly better or different sensitivities depending on what cell type and what experiment it was being done in. The basic gist of the data seems to be from this that the more transcriptionally active, the higher number of genes detected in a sample, the lower sensitivity you expect in terms of detecting these artificial ERCC positive controls that were spiked in. This is an example of some really interesting data analysis that is done with a software package called Seurat that generated from the New York Genome Center. And you can see how going in a stepwise approach, you have plus or minus a treatment, in this case, a drug treatment, and then you cluster them. And initially, those cells that are in one group, the red group versus the blue group, kind of cluster differently. But if you do a dynamic alignment of group 1 and group 2 on, and you get this aligned data set, you can see the distinct clusters and then where the treatments are within each of those clusters. And that can really help clarify the impact of a treatment. And the other thing that's really neat about this Seurat package is they also demonstrate how to use it to compare similar cell types or similar tissues across different species. A number of years ago, when I was doing single cell genomics, the number of reads that were mapping to introns was seen as a problem. And one of the neat things that I've come to learn about since then is that this is not always a problem, but it in fact can be another valuable source of information. In this case, they're looking at the ratio of unspliced reads to those reads for a particular transcript that are fully spliced. And the proportion that are unspliced are typically the most recently transcribed. They haven't been fully spliced yet. And so if you look at that proportion over time, it can be a leading indicator of what's going to happen. So on all of these gene examples, you can see that the blue line trends up or down ahead of the spliced portion, which is the yellow line. If you take that and you overlay it onto a t-SNE plot, you get one of these RNA velocity diagrams that has arrows that show each of the cells, each of the dots in this graph and it also shows where it's going towards, not just what it is at this one timestamp, but where it's going eventually. So let's take a look at a couple of my favorite examples from the recent literature and use those to think about how it is that people are using single cells. One of the ways people use single cell technology is like this paper, where they looked at the development of normal tissue and then they compared that to the gene expression profiles that are seen in tumor growth. And this shows a lot of possibility for identifying new therapeutic targets that are the other genes involved in normal development that when they are misregulated or misexpressed can lead to tumor genesis. Another very common approach to single cell experiments is the discovery of new therapeutic targets that are cell types. This is looking at the lung or the airway and explicitly for cystic fibrosis. So here they actually discovered an entirely new cell type, this really minor set of cells that have a box drawn around them in the scatter plot. They called ionocytes, and they seem to be really heavily implicated in cystic fibrosis, as opposed to an older theory that pointed towards ciliated cells being the root cause. Another cell type example is this paper looking at kidney. So here they looked at nearly 60,000 kidney cells using a 10x platform, and they found two novel types of cells. But what I found more interesting about this paper is when they looked at the gene expression and they compared the different cell types, the different clusters of cells they got, and which genes were being expressed in those. When you look at that and you cross compare it with a list of the genes that are known to have dysfunction in various monogenic or complex diseases, and you can see which cell types are most likely to be important if you're looking at each of these individual individual diseases. So this can really help narrow further study. Another thing that you can really get a good look at with single cell approaches is the differential response of a heterogeneous sample to drugs. This came out of MD Anderson and looks at both the DNA and the RNA of cells and how they respond after frontline chemotherapy, in this case for breast cancer. And so they had a number of interesting findings here. They found, some tumors respond very well and the populations go down uniformly. But in some tumors, especially heterogeneous ones, some clones are eradicated or become extinct even, and others persist. And the total portion of a tumor that is present, of a clone that is present in a tumor, will actually grow. They used DNA and RNA to look at, were these pre-existing very rare cells, or were these cells that are different, that arose in response to that chemotherapy? The answer ends up being that it's some of both. But regardless of whether it was pre-existing or something that arose in response to treatment, they were able to get some guidance on patient stratification based on the gene expression profile. There were some very characteristic profiles that were representative of those tumors or those clones within tumors that would respond positively to this particular chemotherapy. And when there are clones that persist and even expand, looking at the gene expression profiles can help you understand what therapeutic strategies would be a good second course of treatment. If we look in another field of biology, we can see an example where very few cells in the total population are driving a very large response. So this is looking at viral infection and how it relates to individual cell level and viral load. Here, just the yellow cells in the bottom right-hand corner of the scatter plot are those that have significant flu viral particles being generated. Another look at it shows a little more clearly to my eye that less than 15% of the cells are generating more than half of the total viral load being detected. So you've got something that is not being expressed everywhere, but some places it's being expressed very, very highly. The other thing that people get really uniquely from a single cell approach is a paired alpha and beta strands from a T cell, or paired IgH and IgLs from B cells. A number of years ago, this was a pretty difficult thing to do, to compile all of your RNA sequencing reads and then assemble the VDJ region. But now with some new kits from 10X, you can actually do this in a very straightforward manner using their system. And you can use that to say, this particular clonotype, here they're indicated with purple dots in each of these frames. You can not only see who they are by their clonotype, but what they're doing. Are they actively proliferating? Are they effectors? Are they naive? And if you've got a complex immune system, it's got to be much easier to understand what's going on if you can both identify and see what actions they're doing. A lot of work is done in stem cell differentiation, or iPSCs, but that's a low efficiency event, the transformation of regular cells into stem cells and then differentiating those into neurons. Using the Bio-Rad DD-seq, this Sarkar et al. Were able to identify a lot of the things that led to successful reprogramming, and then tied that back also to disease mechanisms for schizophrenia. So that's how people are using it. But I also wanted to highlight for you what I see as some of the most exciting emerging applications, maybe things that you may be able or may be interested in applying to your research. One of the approaches that I'm really high on is actually kind of a general approach called combinational barcoding. So here you split your cells out, either just using wells or a lot of times using fact sorting, and you pool or you color, you code each one of those individual splits, and then you mix them back together and you split them again. And so in this split-seq paper, they did 2 split and pools, and they were able to identify tens or even hundreds of thousands of individual cells by the unique combinations of barcodes. And what they did was use that to identify different biomarkers for all of these different cell types in neural cortex. A very similar approach is called SciSeq. So it also uses combinatorial barcoding, but this is a more flexible chemistry. People have actually used this to do RNA-seq or ATAC-seq, or even look at DNA. So this is a very nice approach. And and here is some of the data from looking at ATAC-seq ran through combinatorial barcoding. The other couple of approaches that I think are really exciting, and we're just starting to see them be applied, use antibodies for protein detection, but rather than conjugating those antibodies with fluorescent dyes, they're conjugated to a nucleotide barcode and then an artificial poly A tail. And so with this type of a antibody that's labeled in this way, you can do your staining of your cells and then detect it using a next-gen sequencer. So here, you don't need much sequencing at all, only 100 reads per protein per cell, which means that if you've got 10 proteins, you only need 1,000 reads per cell. So you can do a huge number of cells on even something like an iSeq, the newest, lowest throughput sequencer that generates 4 million pairs of reads. If you take that same antibody that's conjugated with nucleotide and you pair that in the same experiment, in the same droplets with whole transcriptome, you can actually combine protein detection and whole transcriptome detection in the same cells at the same time. And this Stoichius paper is the first reference. And here they do a nice comparison that even shows looking at the RNA and comparing it to detection by flow cytometry. You can see sometimes they agree very, very well. And then other times the RNA is got a little bit different expression profile. And sometimes the protein is actually hanging around longer than the RNA. And so this gives you both the ability to look at the RNA and the protein, and by comparing the ratios of them, you can see how that regulatory piece in between the RNA and the protein is impacting it. Those two methods looked at using antibodies to look at what proteins may be differentially gene expressed. But if you take those same antibodies and you use them to identify proteins on the cells that are ubiquitously expressed, you can actually do what's called cell hashing. And this allows you to uniquely identify, say, different samples A, B, and C before you put them through a droplet generator or a microfluidic device. And what that'll do is it'll make detection of doublets extremely easy. And this can be really useful for quality control in your experiments. So there are a number of more traditional approaches that looked at proteins, and those had really high cell numbers. But they were really kind of limited in terms of how many proteins you're able to look at. And AbSeq allows you to look at really comparatively high numbers of proteins. The RNA approaches in this purple segment in the middle, they allow you to look at all of the transcripts. So the feature number is really high. And we're just now starting to get to the stage where hundreds or thousands or even tens of thousands of cells can be analyzed simultaneously. And with CiteSeq and ReapSeq, you can actually combine both modalities at really high feature numbers. And through the advent of the more high throughput single cell methods, you can actually look at tens of thousands of cells at the same time. The last application I wanted to talk about is what is often a follow-up experiment. After you find a gene that's of interest, a lot of times you want to perturb that gene and see how it impacts the phenotype of the cells. And one way that you can do that in a single cell level is what's called perturb-seq or crop-seq. In this approach, you take guide RNAs that are used to target CRISPR gene edits, and you attach a barcode to them. And then you do this CRISPR in droplet form. And so individual cells will get either one or two or multiple or no perturbations. And you'll be able to detect that by sequencing the barcode that was on the guide RNAs. By comparing the gene expression profiles with all of these known perturbations, you're able to then look at when this gene gets knocked out, it affects the entire network in this way. And when another gene gets knocked out, it affects the entire network in another way, perhaps. So there are a lot of different single cell approaches. You can use droplets or microfluidics, you can do FACS, or micromanipulation or microfluidics. But all of them, across all of the different modalities, DNA, RNA, and combinatorial multi-omic approaches, can all be detected by sequencing on an Illumina platform. But how much sequencing do you really need? One way to look at that is these sorts of plots that are called rarefaction plots. So on the bottom, you see the number of reads that you apply to each cell. And going up on that vertical axis, you see the number of genes that are ultimately detected. You look at an experiment where you have 1,000 cells and you want to look at 8 different treatments or 8 different samples. If you do 50,000 reads per cell, that is going to be a really good match for a NextSeq 500 flow cell. That'll generate about 400 million total reads. If you take that same read depth, so what you're going to do for something like a DD-seq or a 10x chromium is 15 to 50,000 reads per cell. But you want to do a lot more cells, 32,000 cells in your experiment. That actually ends up being just about the right demand for a NovaSeq S1 flow cell. It's 1.6 billion reads in total. Maybe you recall during the presentation I mentioned in SplitSeq, they did 2 split and pool steps. In that paper, they call out that if you do 3 split pool steps, you are actually able to uniquely barcode well over 1 million cells. So if you have 1 million cells and you sequence them at that same depth, 10,000 to 50,000 reads per cell, you could use up to 10 billion sequencing reads. A few years ago, that would have been a very large project, but now that can be accomplished in just over 32 hours with a NovaSeq S4 flow cell. This graph shows individual dots color-coded by what technique they used. And this is all of the papers that are in this presentation. So you could see, low throughput, maybe something like AbSeq or a manual isolation would be a good fit for one of the benchtop sequences. But something like split-seq or size-seq or the really high throughput droplet-based approaches, it may be a better idea to consider using one of the NovaSeq flow cells to do that experiment. So that's everything I wanted to talk with you today about. I appreciate you giving me your time, and please reach out to us at Illumina if you have any questions. Thank you.
Thank you, Mr. Hoffman, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is, how many cells do you need to include in your analysis to have confidence in your results?
Unfortunately, that's not a very straightforward answer for all cell types, because the number of cells you are going to need is really going to be driven by how diverse the subpopulations are. My approach is that I want all of the populations that you identify, even the most rare one, to include at least 3 to 5 cells. So if you're dealing with something that a particular subtype or a particular rare cell is only 2 to 5%, you need to get at least 100 high quality cells sequenced in order to get 3 to 5 of them. If you're dealing with more rare cell types, obviously the numbers of cells you need to analyze are going to go up in scale as well.
And how do you know what is the correct read depth for these different types of library preparation approaches, such as 3N counting and full-length cDNA?
So some of the papers kind of give you some guidelines, but again, this is one that you really kind of need to look at your particular cell type, and usually the first experiment will kind of point you to the correct read depth. It's pretty common to see 3' N counting approaches used between 15,000 and 25,000 reads per cell. And what you can do is you can actually generate one of these rarefaction curves for your own data by taking the total reads and then artificially downsampling. You can generate one of these curves and see where it flattens off for you. In comparison, if you do a full-length cDNA approach, many of these experiments are done at about 1 million reads per cell, but we just had a paper the other day that I read that said they were only able to get complete segmentation or clustering separation with more than 2.5 million cells. So it's going to depend on how diverse your cells subtypes are and which technology you use. But do a rarefaction curve after your first experiment is a good approach.
Okay, and for our last question, what are the differences between the Bio-Rad DD-seq and the 10X Chromium? They both appear to be very similar in their methods of cell singularization.
They are. They're both, droplet-based approaches, and some of the differences you've seen actually are based on the size of cells The Biorad, I guess the plumbing is a little bit bigger, so you can actually run some larger cells through the Biorad than you can with the 10X. The other thing that seems to be kind of trending out is if you go with a little bit higher read depth, a lot of the places we've seen where they do similar experiments on both, you get a higher number of genes detected using the Biorad chemistry versus the 10X. But there's a trade-off because the 10X gives you gives you more cells, more like 4,000 to 5,000 cells per experiment versus usually in the neighborhood of 1,000 for the Biorad. So another one of these trade-offs between sensitivity and throughput.
All right, well, I would like to once again thank Mr. Hoffman for his presentation. I would also like to thank Illumina for making today's educational webcast possible. Now, before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.